class?
Okay, sir.
Okay. So, but Rohit, very poor attendance, 50%. Total student, I think you are 89, is not it? Or
99, sir.
90? 99. You see, not even 50%. It's very bad. Very bad. Anyway, today it will be the last class, okay, on my part. So first, if I get disconnected, Rohit, you just call me on mobile.
Okay, sir.
Okay. So today we'll just study that is principle and methods of plant disease management. What are the principle and methods? So there is no clear-cut very much differences in both the term. Okay, principle and method. So in plant disease management, okay, mainly it is okay. It is on your in plant disease management. The principle and methods is broadly based on three aspects. One is your cultural practices. Second one is biological control. Third one is chemical control. Okay, and it comprises the integrated management, disease management practices. Okay, beside this, your resistance, that is principal method of resistance, that is by cross protection, by induced resistance, by systemic acquired resistance, also disease management is done. And the last one is your quarantine. That means by putting the legal restriction on the movement of infected plant and plant parts from uh, one place to the another place, or maybe from one region to other region, or one country to other country. Quarantine, this is the legal method of management of plant disease. But in practical, okay, generally when we are talking about the management or integrated management of plant disease, okay, it include cultural practices, biological control, and chemical control. Cultural practices means by our uh, know-how or the knowledge based on the cultivation practices of our ancestors, okay, or presence of mind, your ethic, it is based culture practices. Then biological control, it includes that is use of one microorganism against against controlling the another microorganisms, like pseudomonas or trichoderma used for management or for controlling or for killing the different microorganisms or the pathogen causing disease. Then your chemical control, here we use different fungicide, antibiotics, etc. to seize the growth, development, or to kill or to retard the activity of microorganisms. Okay, so starting with the cultural practices, number one is site. The first one is site selection. So it should not be sick plot. That means the cultivation should be avoided, or raising of crops should be avoided in the sick plot. That means sick plot in case of your soil-borne disease. If it is known that a particular plot, suppose your damping of disease, it means the death of the plant just after the emergence, seedling at the seedling stage only. Okay, so if there is some plot like that, if some seed bed is made or seed is sown, so everywhere, every year or every season, suppose if damping of disease appears, or in a particular area, if a particular crop is sown, so if the wilting disease or vascular wilt appears or some root rot appears, that means that plot is sick. Sick means it is having the lot of microorganisms or propagules, or it is harboring the your resting structure of the pathogen or the pathogen. So this is known as sick plot. So sick plot is not desirable in your general cultivation practices. Okay, but this sick plot is of high importance in your science or in research. How? Can you tell someone? Sick plot in general cultivation practices, it is your not desirable. Okay, one should, one should not take the sick plot for cultivation, but in research, okay, or in doing the science, it is of great importance. How? Why?
Sir, it provides optimum condition for different growth,
Yeah.
different microorganisms.
Yes, yes, correct. You are right. So someone further will add something like Rajiv Ranjan Minal, Puspanjali, Ayush Gupta, Preeti Kumari, Aditya Raj. What the Rohit told, okay, something you can add? Why it is done so?
Sir, it help in the study of nature of microorganism.
Nature, no, no. Why sick plot is needed in research? Why? Hmm? You see, 147. Where the other students are there? 38.
So we can study about the cycle of infection.
No, you see, I told why it is from research point of view it is important. That means in general cultivation practice we avoid sick plot, but in research we need or we create, rather we make the sick plot. Because suppose if you are having the 10 germplasms, accessions, cultivar, varieties, Aniket,
Yes, sir. Varieties, cultivars.
this thing, germplasm.
Germplasm is a collection of all the uh, particular desired traits or genes.
No, germplasm you can simply tell. Suppose in case of pulse, okay, if you go like in farmer's field, if you see some different type of character of pulse, okay, plant type like grain, whatever, you just collect the seed. It will be a collection, germplasm. Then if you go to some other place like your south, okay, from there you can collect. So like this, it will have. So it may be collected from a region, okay, within a district, within a state, outside the state, within the country, outside the country also. That will be known as exotic, okay. So suppose you have collected, then in suppose pulse, okay, fusarial wilt is a problem. It is a soil-borne disease, okay, caused by Fusarium oxysporum, Pharma specialis, Udam. In case of Kajan, that is your pigeon pea, okay, red gram. So you want to see whether you have collected the 10 germplasms of your red gram or this pigeon pea, and you want to see that is whether any one or how many these germplasms are okay resistant to the wilt disease. How will you say? How will you see? How will you see? You have to show this is soil-borne disease. Okay, you have to put in the pots or in the soil. Then you have to add the inoculum or the pathogen, particular pathogen against which you want to screen the germplasm or you want to see its performance. Okay, so you have to put in the pots or in the soil where you are showing the culture of Fusarium oxysporum forma specialis udam. Okay, you have to create the sick plot artificially. Then you have to see that in like suppose you have planted all the 10 germplasms, okay, you'll find maybe one germplasm, maybe two germplasm, or maybe none of the germplasm will survive. Or suppose 8 germplasms have died due to the wilt symptom and 2 has survived. That means these 2 germplasm is having the means the different degree of this one resistance against the pathogen. Unless and until in the soil the pathogen is there, you are showing, okay, how you will come to know that it is resistant against the disease. If there is no fusarium, so you have shown the 10 germplasms of pulses, okay, seeds of 10 germplasm, all will survive. Then whether you will tell that all these are resistant to the fusarial wilt of pigeon pea? Aniket? Rohit? Subin?
Yes, sir.
Whether you will say? Minal? Aniket? Aniket?
Yes, sir. If
You are
all,
not? Yeah, tell.
sir, if all of them survive, then they should be resisted.
No. Yes, means if you are not putting in the pot or in the soil your fusarium, your this one, oxysporum pathogen, then it will survive. Then you will tell it is resistance against the fusarium or the wilt disease.
No, sir. If the inoculum is not present, then it cannot be told.
I will check. Once the inoculum is present there, okay, and if it is able to kill some of the germplasm, or if not, also if there is sufficient amount of propagule, okay, inoculum is there, and in the presence of the sufficient your quantity of inoculum, if this plant survive, that means it will be resistant, is not it? So artificially the sick plot is also created, okay? But in
Sir,
general,
sir, if
yeah.
sir, if the potential of the inoculum is low, then how will tell this?
Now, that you have to see. There are in the already you are I mean, there are stocks varieties. Okay, susceptible check, resistant check. Okay, check means now I am coming to some more high. Check is variety, you can say. Okay, someone is resistant, someone is susceptible. So you have to put both. Okay, and in that condition, you will in the susceptible one will show the disease, 10% disease. That means the inoculum has the potential to kill the plant. And in the same condition, okay, resistant plant will not get killed. Resistant check. Okay. So these are the methods of scientific study. And we both susceptible check, it is known as check. Okay. And resistant check is to be put with the screening or evaluation of the germplasm. Okay. And it is compared, the performance of the germplasm is compared with based on the performance of susceptible one and resistant one. That means if it is towards the susceptibility, how much susceptible? If it is towards the resistance, how much resistance? And the standard is the resistant check, susceptible check. Okay. Okay, Rohit. Aniket. Means if not potential, that means the susceptible check will also not get disease. Is not it?
Yes, sir.
Yeah. Clear?
Yes, sir.
Yeah. Coming to our point, that is sick plant should be avoided in general cultivation practices. Also, low-lying areas with poor drainage should be avoided. That in low-lying areas having poor drainage, okay, will have your accumulation of your moisture, excess moisture, and it will just create a climate, okay, or conducive soil condition where this soil-borne pathogen will have its maximum activity. Okay. Then also the close spacing, it will not come, it will come later on. Here site selection is okay. Then example, avoiding such areas lowers or minimizes the chances of soil-borne diseases like damping of and root rot diseases. In vegetable, particularly solanaceous crops, as I told, okay, mainly vegetables caused by species of Pythium, Phytophthora, Fusarium, Rhizoctonia, etc. Pythium, Phytophthora, this belongs to this your Omycetes, okay, and you will get Fusarium or like Pythium, Phytophthora, Fusarium, it causes your weight rot or damping off, or but in the Rhizoctonia, it causes the dry damping off or dry rot. Okay, that means in the absence, in the scarcity of water, the damping of symptom appears, okay, as in case of the excess moisture. But here the pathogen responsible is Rhizoctonia. Second is fallowing. That means keeping the Fallowing means without cultivation, you have to keep the land, you have to plow it down, and the soil should be exposed to the bright sunshine so as to kill the your resting spores or the vegetative structure, whatever is present in the soil. Okay, it gets killed by the bright sunshine, or by the mechanism of mainly the desiccation. So keeping the cultivable crop uncultivable from crop and weed, it is done to reduce the inoculum load. Example, by soil solarization, inoculum load of Xanthomonas campestris pathogenesis, okay, causing BLB of rice can be reduced. Flooding helps to reduce the incidence of fusarial wilt of banana and root knot nematode incidence in vegetable crops. How, then question comes, how this flooding helps to reduce the incidence of this is fusarium and the nematode, root knot nematode. How?
Lack
How?
of oxygen.
Yeah, that means it creates your apophysis. That means anaerobic condition, and these organisms needs, okay, particularly this root knot nematode, okay, it needs the oxygen for its activity, for respiration. Like fusarium is also anaerobic. It is not anaerobic, it is aerobic. Okay. So due to the flooding, all the pore space of the soil it is closed. Then plain seeds. It is done by drying. Okay. Also, some burning seeds in 20% initial solution. Okay. Nematode seeds, cockles, fruits, and it is being by hand. That means in case of your lose water, okay. Then here one photo, system one photo, the seeds prior to the sowing, it should be also implanted in a line solution in the okay. Then the infiltrated seeds that they are lighter than where they float, okay. And then this is floating seeds can be decontaminated and discarded prior to sowing. Example, why is the super? Okay. It's called by your alveolar candida and system okay. Such diseases, skin-borne diseases can be checked by this method. And at the sowing day, sometimes early or delay sowing and available the pop-up. We see a critical period of disease in them. So for critical period, something usually the early stage, some will the vegetable stage, some will the reproductive stage, some will the early stage, like that. Okay. So your by this showing day, we can avoid the we can avoid the time, okay, which is that suppose the all animal condition it is very much favorable during your our like it our last support. So and at that time if the accident started is in the immature stage, then your alternative guideline may occur. So what you have to do that you so after this is 15th of October earlier, that when the plants get hard and early stages crops, then the certain area will not be able to cause the disease or the this is an example of this. So early delayed sowing and available crops get the disease. Example, five mustard, okay, alcohol red causes more than the crop is sown late. That after mid of October, if the crop is sown mid of October, so there is more incidence of white mustard. Sorry. Like because there's the crop was sown early. That's what the crop was sown. So early, let's somewhere early. So this is white mustard is okay. Negligible. Okay. by adjustment of showing that you can just escape the critical problem and the disease occurs. That means to avoid the time when the pathogen is very virulent and at the stage at this time, the stage of the crop is very vulnerable for infection with the pathogen. So we can just adjust the showing to just shift the vulnerable stage or the susceptible stage of the crop if you go after. Okay, when the pathogen is very virulent. Crop rotation. It is useful in insects and modern pathogen. Okay. Mainly, also is practiced, but it will be limited extent. Okay. So it is basically that you may require of food the pathogen resident in the soil. Mainly, this crop rotation is done against your soil borne diseases. Crops are selected between the susceptible source of crop should be resistant or immune to the particular pathogen. That means that the crop which is selected, okay, should be immune host, okay, of that pathogen should be resistant immune to the particular pathogen. As an example, your incidence of growth available can be modified by crop pathogen. Paddy and wheat, because the vegetables are associated and this paddy and wheat affects your granularity immediately. And this crop rotation is actually general if it is just for 2 to 3 years. By destruction or by microbials. Destruction or by destruction of pathogen soil is by many mechanisms employed by antibiotics. Rohit, Aniket, it's okay.
Yes, sir.
Aniket,
Yes, sir. Yes, sir.
Control. Then destruction, I told the destruction or survival of pathogen in soil is by mechanism employed by antibiotics. Here, consumption for space and nutrients is the principle. Here, the consumption for space and nutrients by agonists with pathogen for the same energy source. That means suppose if iron is needed by pathogen and whatever the you are adding, so you have to put the pathogen stronger than the pathogen in taking the iron or the your ferrous compound. Okay. So that the pathogen it will take care of the ferrous compound and make it not unavailable to the pathogen. So since it is needed by them, so instead of the activity of the pathogen will get retarded or may get killed also. Okay. The production of the enzymes such as gluconeogenesis and pathogenesis to lyse fungal cell wall. So here also the pathogen which release enzymes, which are responsible for regulating fungal cell wall. Then this pathogen also produces antibiotic and inevitably metabolites production. This metabolites they are producing secondary metabolites like hydrophore, then different antibiotics. What is hydrophore? Yeah. From money you are dealing. Jivan Jyoti. Amar Jyoti. Amar Jyoti. Amar Jyoti. available food and then mother is fine anyway. So this is also hierarchy letting compound. Okay, they will bind the iron compound and will make available food. Like they are producing different like pyridine, nitrine, okay, which are very from the pathogen. They are produced by the bacteria like Pseudomonas, like controllers and Pseudomonas fluorescens. Then pyridine or parasitic action against the pathogen, modification of the environment, and further the pathogen. Like okay, it is a parasitic action. It just make fighting effect around the pathogen might. Suppose there is there or your compromise is there. So this is what might use. So it will just while the fungal mycelium and it might it cannot allow you to grow further. And some regulatory applications also there. It is mainly a case of entomophagus fungi. Okay. There are some fungus. Okay. Which are just kills the insect. Example, like already control lithium cause in tomato. So there are a lot of examples of where this very effective by pathogens. Then there are no problems. That if you are using a pathogens that which will end the formation of so automatically the pathogen will and it's check. It's not it. With multiplication and colonization of it favors the development of pathogens on area plant parts where the population and then that such pathogen they will survive and antagonism means these are the or antagonistic effect on the pathogen. It is done by using big one trap and let's define them both and then we will use five. You I trap up and then the normal purpose it is grown so normal get affected so very good plant. So the very good plant updated and you get right. So it introduces lower down population of normal soil. Example like fluorescence inhibits reduction of militia levies. It causes the compound one okay. So this is fluorescence by okay. And inhibit the formation of the spore okay. Which is responsible for the common one topic. The reduction of virulence of the pathogen. It invades the section of the pathogenesis different pathogen. That means there are five pathogens which is just lower down the activity of the pathogen. Okay. Means how suppose there are some tips, some heights, okay, and in the some comes their activity comes less. So like that. In the presence of a pathogen, the activity of the pathogen gets okay, and gets your down or gets lower down. Okay. As a case of arrival of police. Okay. The activity of notorious elements gets reduced. It can be achieved through modification of soil environment and factors inherent in the pathogen. It also determined by the availability of another environment, and it is mainly applicable or it is seen in case number two. Example, insufficient production leads most larvae to become damaged in case of the active data. It leads to
आपकी आवाज नहीं आ रही है सर।
Hello, hello.
Yes, yes, sir.
Someone just call came urgently. That is why the connection is gone. Anyway, slide is not visible.
No, sir.
Just a minute. I will make it visible. Now it's visible. Okay. So this is all about your cultural control. Okay. Then we are coming to the no, where we are biological control is not it. Um, Reduction of virulence of pathogen. Then fourth one is your biological protection of planting material. So it is the most successful protection of planting material. Then more mass incorporation. So uh, it is most successful if the planting material is treated with the biocontrol agent like root dip treatment or priming of the root is done, okay, with a solution of biocontrol agent. So it is more effective rather than the biocontrol agent. It can be achieved by protecting the covering of seeds, rhizome, tubers, and fruits with propagules of antagonist. Okay, so if some coating is made with the biocontrol agent, maybe it's rhizome, tubers, fruits, okay, with the propagule or with the, that means your propagule, the biocontrol agent, it is very effective. Seed coating with cell or spore suspension of antagonist enhances their number in the rhizosphere. So if a seed coating is done and when it is sown, so the number of your biocontrol agents or antagonist increases in the soil, okay, in the rhizosphere. Rhizosphere means the soil covering the root zone of the crop. Okay. So the seed coating, the priming of seedlings or roots is done with the your biocontrol solutions of biocontrol agents, okay, made of pseudomonas, trichoderma, bacillus, or sometimes consortia, okay, consortia of these biocontrol agents. Consortia means in that solution more than one type of biocontrol agent is added. Like suppose pseudomonas is also there. Suppose in a 1 liter, if 20 gram is to be put biocontrol agent, then 10 grams may be bacillus and 10 grams may be pseudomonas. Okay. Then biological protection of foliage and blossoms. Here the epiphytic flora suppresses pathogen through competition for nutrition and space, antibiotics, and hyperparasitism, or through induction of in host. Okay. Example: strawberry gray mold rot caused by botrytis cinerea. Okay, it is its incidence is reduced by 90% by the application of glycladium rosium. Okay, this mold disease incidence can be reduced by application of your culture of glycladium rosium by spraying with the culture of glycladium rosium. Then lastly, the chemical control. It is the most effective, but nowadays it is discouraged because of its residual impact, okay, or residual effect. Some chemicals act by inhibiting the activity of pathogen to synthesize certain of its cell wall substances by like acting as solvent, damaging cell membrane of pathogen, forming complexes with and thus inactivating certain essential coenzymes or enzymes of pathogen and thus causing general precipitation of protein of pathogen. Example, wettable sulfur. Okay, in this the wettable is sulfur interferes with the electron transport along the cytochrome system of the fungi, thereby depriving the cell of energy. I think in the last class already you have read, I studied the mode of action of fungicide. Okay. And second one is the denaturation of enzymes. In Bodo mixture, okay, copper, it reacts with the sulfhydryl group of amino acids, okay, which are the causes of enzymes. Okay, that means it just restricts the formation of or it retards the activity of enzymes, okay, needed by the pathogen for its activity. Rohit,
Yes, sir.
okay. Aniket,
Yes, sir.
mode of action already I have given in last class related to that only. So third, like many organic chemicals, means fungicides, okay, inactivate proteins and enzymes through reaction with their SS group. Okay. So example, dithiocarbomates, when taken by fungal cells, releases carborhyl. PCNB, it is a fungicide that is pentachloronitrobenzene. It reacts with your ammonia and SS group and inactivates enzyme that have such groups. Okay, that means enzyme inactivation is when this fungicide is applied in the soil. This PCNB is applied in the soil to check the activity of your clubroot disease of crucifers or cabbage. Okay, cauliflower caused by fungus Plasmidiophora brassicae. Then several systemic fungicides inhibits ergosterol biosynthesis. Ergosterol is a cellular compound that plays a critical role in structure and function of membrane of many fungi. So, some of the fungicides, it will just what will do, it will inhibit its biosynthesis, like your imazeli. Some nematicides disrupt the function of membrane and example, hydrogenated hydrocarbons, methyl bromide, soil fumigants. This will disrupt the fungus. This will disrupt the function of membrane and nervous system. Okay. Whereas other inhibit the nerve transmitter enzyme cholinesterase and causes paralysis and death of nematode. Example like ketazine. Okay. There is organophosphates. So this will disrupt the function of membrane and the nervous system also is disrupted. So ultimately the pathogen is not able to do its routine work or the work related to pathogenesis. So in turn your disease activity or the infection activity by this pathogen, okay, especially the nematode is checked. So this is all about principle and methods of plant disease management. Anyone, any doubt, any student?
How fungicides act. Sir, can you tell me how seed protectants act?
Oh, okay. Seed protectant, okay. It is done with the two things. One is maybe with chemical or maybe with the biocontrol agent. Okay. So when it is seed is protected with the fungicide like the therum, captan, okay, or a mixture of therum or carboxine. So it will just make a coating, okay, seed around the seed, and it is air dried, then it is sown. So the fungus present in the soil, okay, when it comes in contact with this seed, they are not able to perform its activity. Okay, it repellent very principally. After that, suppose even if some fungus comes in contact, they get killed, okay, by different mechanisms, like, like your uh, disruption of fungal cell wall, okay, okay, disruption of fungal cell wall, it will just disrupt due to the activity of these chemicals, okay, or these chemicals also produce some slow releasing compound which is deleterious to this pathogen, okay, particularly fungus or maybe the bacteria. This is one part. Again, if the seed is treated with the biocontrol agent, if it is coated with your a solution of Pseudomonas, Bacillus, or cocktail or trichoderma, or upon putting in the soil these seeds, these biocontrol agents also gets your multiplication in the soil in favorable condition. And when they will multiply, if trichoderma is there, it will do the mechanical, through its mechanical coiling effect, it will bind with the pathogen and it will restrict its movement. Suppose if the biocontrol agent is used like Ralstonia and Pseudomonas fluorescens, so it will produce some siderophores, which in turn will bind with the iron compound and make this iron compound unavailable to the pathogen. Okay, in that case, is food and nutrition of the pathogen will be hampered. Okay, so its activity will be this a biocontrol agent like your Bacillus, Pseudomonas, it produces secondary metabolites and some antibiotics like pyronitrin, pyroviridine, which is having the inhibitory effect on the spores or the vegetative structure of the fungus, okay, of different pathogens. Okay.
Yes, sir. Sir, another question.
Yes.
Sir, for crop rotation, that it is only viable for 2 to 3 years.
Crop rotation.
Sir, if
viable,
yes, sir.
it should be done at least. It should be done.
Yes, sir.
Yeah.
Sir, if there suppose there is a land and in that land there can be spores of different pathogen which have different period for different viability periods. Some can be some can like cause disease in
Yeah.
2 years or some can do in 4 years. So how can we confirm that?
You have to do, suppose you have to study what kind of pathogen is present. Okay, like your this one, Pythium phytophthora. If it is present there, so crop rotation for 2 to 3 or Rhizoctonia, crop rotation for 2 to 3 years. Okay, if fusarium is also there, like your annual crops or like your cereal crops like pulse, it can be also controlled 3 years. But the Fusarium oxysporum, Forma specialis, your CD, causing the wilting guava, it survives for long, more than 10 years, by forming the chlamydospore at the very lower depth. So if in some places, if the incidence of your guava wilt is there, so here the Forma specialis CD, okay, it survives more than 3 years. So there the crop rotation with the non-host crop, okay, that means you should not go for planting of guava. With some other crop you can go for planting. Number one. And if even if the guava is to be planted, so you have to wait for at least 10 to 12 years. Okay.
Thank you, sir.
With non-host crops, crop rotation should be done with non-host crops. Okay, to get healed. Because it is not like that this time you are growing the pulse. So you want to just minimize the population or kill the Fusarium oxysporum. So only 1 year, if you don't plant the pulse, you put the suppose paddy, it will get killed. Not 100% will get killed. There will be among 100, maybe 20 will remain there. Again, after 1 year, if a pulse you take, again it will multiply. So for its complete or 100% or to get its population below the desirable level, at least 2 to 3 years of crop rotation is needed. Number one. Number two, from the practical point of view also, crop rotation cannot be done for long because crop rotation is under the pressure only. Otherwise, if you are one is doing the vegetables or pulses, that means it is the profitable crop. Then only one is growing. So it is done only under the pressure. So under pressure, how many years one can go for crop rotation? Okay. Okay, Aniket, who was telling, who's query?
Sir, how organic soil amendment help in control of plant pathogen disease?
You see, organic amendment, what happened? Organic amendment, when you are doing, it just has the volatile effect. It emits some volatile gases like your hydrogen sulfide. And if this use organic amendment, it also increases the activity of your beneficial microorganisms present in the soil. Okay. So when the activity of beneficial microorganisms in soil will be increased, so definitely it will take care of the pathogens present in the soil. Okay.
Yes, sir.
Yes, Amar Prem, tell.
Yes, sir.
Anyone, any question? Anyone? So should we move to the next? That is pathogenesis. Last chapter. Aniket,
Yes, sir.
should we move? Can we move to the last pathogenesis?
Sir, can you please take it
Pathogenesis.
in next class?
No, there is no time next class. Today is the 2 period. Why you are telling like this? Today is the 2 period, na? Up to 5.
Yes, sir.
Then why you are telling like this? Why? What is the problem? Is there any problem?
No, sir.
Then, because examination is again coming, lot of thing is there. Just finish and enjoy.
Okay, sir.
Okay. Only it will take hardly your 15-20 minutes. Okay.
Yes.
Okay. Pathogenesis. So pathogenesis, that means it is the process. Okay. But that is the process. That is how the pathogen causes your infection or disease to the crop plants. Okay. The first one coming to very straight way, infection. So infection, it is the process by which pathogen establish contact with the susceptible host or cells or tissue of the host and draws nutrition from them, okay, or from there. So infection is the first process, that means landing of the pathogen on the susceptible host plant. And for getting its establishment, okay, and drawing the nutrition. So during infection, pathogen grows or multiplies or both within the plant tissue and colonize the host. That means it will grow, it will multiply, and side by side it will colonize inside the host. Thus, invasion and colonization are the two concurrent sub-stages within the stage of infection. Invasion means just penetration, or you can say attacking, and colonization. That means after entering or after attack, your growth and multiplication. These are the two concurrent sub-stages of infection. Successful infection results in the appearance of symptom. If there is successful infection, so there will be definitely appearance of the symptom will be there. In case of perennial crop, it takes more than a year after inoculation. That means perennial crop, like in your tree crops, like suppose coconut kadang-kadang disease, it is caused by coconut kadang-kadang viroid, and it is the smallest viroid known to cause the plant disease. So to get the symptom expression or to disease, at least it needs 4 to 5 years after the inoculation of the pathogen in the host crop. But like your lead blight of potato caused by Phytophthora infestans, okay, just after inoculation, within few days, the expression of the symptom is there, and it causes the disease to the crop. During infection, pathogen releases enzyme, toxins, and growth regulators in host that affects structural and physiological integrity of the host. In response, host reacts with a variety of defense mechanism. That means when the pathogen attacks a host, okay, so this pathogen also releases enzymes like your cellulases, which will degrade the uh, cellulose of the cell wall of the host plant, or it releases pectinases. It will degrade the pectinase of the host cell. Some toxins also produced like fusarium, produces your fusaric acid, which is toxic. Okay. So, in response to these activities of the pathogen, the host also uh, had some structural and physiological integrity of the host. Okay. Uh, so this host, it like it is having the different your structural defense mechanism as well as biochemical defense mechanism. So this structural defense mechanism may be your uh, pre-infection defense mechanism structural and post defense also. Pre, like suppose in case of some crops, okay, it is having the spines, thorns, its surface waxy. So whenever a pathogen lands on it, okay, it falls away. If the surface will be waxy, so along with the water droplet or something, if it falls, or as such, it will not um, can get cold on the host surface in case of occupantia plant like that. Okay, or sometimes it is the plant surface have the waxy layer, so the pathogen is cannot penetrate inside it or cannot easily penetrate inside it if it is the your waxy layers there, cuticle layer is if it is very thick. So these are the pre-infection defense, structural defense mechanism by the host. Then post structural defense mechanism, once the pathogen gets entered, so the host surface it forms different structural your modification inside it, like formation of cork layer, okay, tyloses. That means it will just uh, kill the pathogen and it will just restrict the movement of pathogen beyond a particular point or from the point of infection by forming the dead necrotic area in the host, by forming the corky layer. Okay, tyloses, that means tyloses, it just restrict the movement and spread of vascular pathogens inside the vascular system. Okay, it is net like a structure. Okay, it will restrict the movement and the growth of the fungal structure inside the host system in the vascular vessels. For a successful infection, the disease tetrahedron must be fulfilled. You might be knowing the disease triangle. You know disease triangle? Disease tetrahedron? Disease pyramid?
So
Yes?
disease triangle.
Yes. Tell, Aniket.
So host, pathogen, and environment together form a disease triangle, whereas in tetrahedron, a fourth element, human, is also added. The intervention of human.
Is it?
The triangle.
Disease pentagon, a disease pyramid. Even this disease triangle, someone can explain with some precise term or nicely. Disease triangle. Anyone? Is it mere the host pathogen environment or something else? Amar Prem,
Yes, sir.
is it simply the host? Yeah, who is?
Sir,
It is interaction between the environment, host, and infection, which is majorly a biotic agent, sir.
Okay, but still,
sir,
something
it shows, sir, each arm of the triangle shows different thing. Some like the pathogen side shows the virulence or the abundance of the pathogen, and then the for the host side it shows the susceptibility, and for the environment side it shows
okay,
the
okay,
conditions.
all right. Who is telling?
Sir, Shivanti Das.
From Odisha.
No, sir, West Bengal.
Oh, West Bengal. It is like that. You see, the host should be susceptible, pathogen should be virulent, and environment should be favorable. Okay, as Aniket told, it is an interaction between host pathogen and environment. If even if there is interaction, disease may not be there. Why? If suppose pathogen is not virulent, it will be not able to cause disease. If pathogen is virulent, host is not susceptible. Okay. If the host is resistant, then it will not get diseased. Okay. If host is susceptible, pathogen is virulent, but environment is not favorable. That means for its activity of pathogen, okay, it needs certain range of temperature, moisture, humidity, okay, daylight, wind speed, many things, different meteorological parameters. So if it is not fulfilled, then it will not, okay, cause disease. Now my question, okay, human factor, how it plays human factor, Aniket, role?
Sir, the interference of human or the cropping practices that we adopt may enhance the disease that is exposed to crop.
Or suppose like human activity, if there we see if it is expected that some disease will come, if we can from our knowledge that if such type of environment is there and we have shown if the variety is susceptible to disease may come, they can go for prophylactic measure by spraying of the fungicide and all these things. Okay, but you see if the host is also favorable, pathogen is your virulent, environment is also conducive, even if such thing is there, but there may not be development of disease. Why? Why? There may not be development of disease. Why? Anyone? If the pathogen is susceptible, host is susceptible, pathogen is virulent, environment is your favorable. Even if in this and they come in means their interaction, but even if such condition is there, disease may not be there. Why? Why?
Sir, the inoculum may not land on a particular site that is susceptible to the infection. The point of inoculum landing might not be suitable on the host.
Is able to cause disease. Why?
So your voice is not coming.
सर चले गए
Or
Hello, hello.
Yes, sir.
Now it's okay. So what I
Okay,
was asking, yes, what I was asking that in the presence of your susceptible host, virulent pathogen, and favorable condition, okay, there may not be the disease. So why? How?
Sir, maybe because of less number of pathogen present, the
No. If a
pathogen
number,
are in less
so lack
abundance.
of interaction between them.
yes, you are coming to the point, but little bit you tell.
Proximity of the inoculum from the host.
Yeah.
The
Right.
the
Yes.
proximity of the inoculum from the host.
Now, if it is there is no problem of proximity, then then now slide is okay.
Yes, sir.
Now tell. You see, even if all the conditions are there, but time factor is very important. Okay, they must come in interaction for a definite period of time. Like in lead blight of potato. Okay, it needs suppose susceptible host is there, their virulent pathogen is present there. The condition of environment, that is your foggy weather, very low temperature below 8 degrees centigrade. Okay, Mm. Moisture is there, high moisture, very close planting. But if such type of condition prevails at least for 48 hours, that means particularly the environmental condition. Okay, if it prevails at least for 48 hours, then only there will be a successful disease development, lead blight, the disease development of lead blight of potato. If such condition prevails for less than 48 hours, like suppose 18 hours, 8 hours, 10 hours, okay. So even if in presence of favorable pathogen, the condition was conducive for only 8 hours, the host was susceptible, the disease, there will be no development of disease. Okay, so they must come in interaction for considerable period of time. Okay, so time factor is also very much important. Okay, Rohit,
Yes, sir.
Abhinav. Okay,
Yes, sir.
yeah. Coming to the invasion, okay, pathogen invades in different ways and to different extent in different hosts. There will be way of invasion, means penetration or entering, and to different extent. Like in pathogen, the activity is the superficial or maximum. The penetration of the mycelium is up to the epidermal layer. Okay, but in case of systemic pathogen like wilt and all these things, it just enter inside the host system. It becomes vascular in nature. So fungi causing apple escape and black spot of rose produces mycelium that grows only in the area between cuticle and epidermis. That means very superficial. Others such as powdery mildew, it produces mycelium not only on the surface of the host but send haustoria into the epidermal cells. So little bit depth. Most fungi spreads into all the tissues of the plant systemically, either by growing intracellular or intercellular mycelium. So the mycelium, the growth may be intracellular or intracellular. So you know the difference between intracellular and intercellular? Intracellular means within a cell. Inter means your in between. Okay, from one cell to another cell. Pathogen causing vascular wilts invade xylem vessel. Many infections caused by pathogens are local, that is, they involve a single cell, a few cell, or a small area of pathogen. So, when a small area of the host is infected by the pathogen, so this type of pathogens are non-systemic in nature. Their infection is localized, like leaf spot, leaf blotch. So wherever the pathogen will fall or it will reach, it will have its activity. But in case of a systemic disease like dieback, like your wilt, okay, so these diseases are systemic in nature the pathogen may get entered from the root or some aerial parts or collar region of the plant, but it will spread slowly to the entire plant. In entire plant, the system will be expressed and the plant will get killed. Okay. Infection caused by fastigious vascular bacteria, molecules, protozoa, viruses are systemic. Okay. Some fungi like downy smuts, rust, they also invade systemically. Okay, like downy mildew, it will enters inside the host and the whole plant will get infected. Smuts, then rusts, etc. Then colonization, most of the pathogen continues to grow and branch out within the infected host. That means most of the pathogen, they will continue on growing and they will side by side, they will branch out in the infected host. In some fungi, in some fungal infection, while the younger hyphae continues to grow in the new healthy tissue, older ones already infected area die out and disappear. That means just acropetal in nature. That means the new fungi will go on invading in the host tissue and the old one will get degenerated. Bacteria, molecules, viruses, viroid, nematodes, and protozoa do not increase much in size with time, rather they multiply rapidly. That means hypertrophy. Okay, not hyperplasia, hypertrophy. Oh, multiply, sorry, it should not be hypertrophy, it should be hyperplasia. Hyperplasia is the excessive cell division. Hypertrophy is the enlargement of single cell. Okay.
Yes.
Cancer is your hyperplasia. That means uncontrolled cell division. Your club root of disease, club root of cauliflower caused by Plasmodiopora brassicae is the hyperplasia. Okay. Hypertrophy is the gall formation. Bacteria, most of the fungi produces mycelium only within the host cell and in fact like vascular wilt. So many fungi, they will produce mycelium inside the host and they will infect like the vascular wilt. Sorry. Relatively a few fungi produces mycelium on the host surface such as powdery mildew. Powdery mildew, okay, they will produce the mycelium on the host surface. That is why in last class I told you if there is infection by the powdery mildew fungi, okay, Pernospora, so in management practice, it is recommended to just spray the plant with the water. So what will the incidence of this disease, this powdery mildew get reduced because by spraying by or washing the plant, okay, the fungal structure or spores get washed away. So it also minimizes the disease because the pathogen is superficial on the top surface. Bacteria, viruses, viroid molecules, protozoa produce only inside the cell. Exception is the leaf spot by bacteria. It reproduces outside the host. In case of the leaf spot, like angular leaf spot of cotton caused by Xanthomonas compostris pathogen malvacearum, the multiplication of this bacteria is outside the host surface. Fungus produces several thousands to several hundreds of thousands of spores produced particularly per square centimeter of the infected plant is numerous. From specialized sporophore spores produced on infected plant is often in millions or in trillions because they are the microorganisms. Okay, very fast your multiplication and a number is also very huge. Bacteria reproduces rapidly every 20 to 30 minutes by binary fission. Millions of bacteria may be present in a single drop of infected plant cell. In case of infected plant, if ooze is taken, so in a single drop, there will be millions of bacterial cells. Okay. This is all about your pathogenesis. That means your process in a nutshell. It causes the disease to plant. That is first infection, then invasion, and lastly colonization. Okay. Aniket, Rohit, Amar
Yes, sir.
Prem. Yes, anything? Hmm? So this finishes your syllabus from my part. Anything?
Sir, you told about the disease pentagon. What was it?
Ah, pentagon. That is a tetrahedron human factor is there. Okay. In pentagon, your time factor also comes. Okay. Okay. Aniket?
Yes, sir. Yes, sir. Yes, sir.
So tell anything. You give your attendance in chat box.